Hello, I'm your host, Ray Dogum, and welcome to VibeCast. VibeCast is VibeBio's very own podcast where we explore some of the hottest topics in drug development, investment strategies, and technology innovation. At VibeBio, we're harnessing AI to enhance biopharma's efforts with drug pipeline prioritization, clinical development planning, and advancing treatments for rare diseases. Our guest today is Alex Federation, co-founder and CEO of Talus Bio. Dr. Federation received his Ph.D. in chemical biology from Harvard and invented the MARMOT platform during his postdoctoral fellowship at the Altius Institute for Biomedical Research in Seattle, where he trained in computational epigenomics and cancer biology. Alex, thank you for joining me on the show today. I'm really excited to have this conversation with you. If you wouldn't mind, to get this started, would you share a little bit more about yourself and your background, and sort of what got you into this biotech journey you're on now?
Yeah, thanks, Ray, for having me. I'm excited to be here. That's a great intro. A lot of what we're working on at Talus really goes back to some of those early days in my training. So I was sort of lucky enough to train in a very entrepreneurial environment. I worked with a guy named Jay Bradner, who at the time ran an academic lab at Dana-Farber, but we were really trying to tackle this problem of the undruggable genome even back then. And a lot of the ideas we worked on got spun into companies there. So I saw how it kind of looks, at least in Boston. It's a little different out here on the West Coast, I guess, so when you're starting a company. But I got to see that there. After I left, Jay actually went on to lead research at Novartis, so he was very entrepreneurially minded as well. And training in that environment definitely demystified it a little bit, and also just planted the seed that it's sort of irresponsible to label something as undruggable, especially with all these amazing new technologies coming to the forefront every day. So I came out here to Seattle, met my co-founder, Lindsay, at the University of Washington while I was doing my postdoc out here. And together we kind of came up with a lot of the technology that underlies what we're doing at Talus. And that was sort of the starting point was that collaboration and the two academic labs.
Awesome. And you mentioned something that I think I want to just dive into a little bit. You said the undruggable part of your genome. For people who might not be so familiar with that, can you explain what that means?
Yeah, for sure. So if you look at every drug on the market today on the shelves, they only target about 10% of the proteins in the human biology. These are, people call it sort of the low-hanging fruit. So proteins that essentially, when you take them out of the cell or out of your body, they still work in a test tube. You can use biochemistry and molecular biology to find drugs that block their activity in that test tube, and then sometimes it'll work in a human, and that's how you make a drug. The other 90% of the proteins that are in human biology have been sort of written off as undruggable, either because we can't get access to these targets or their structures are too floppy to fit a traditional small molecule into them. People were saying this the first day I walked into class at Harvard. You go to the fancy building, chemical biology, chemistry building. This is where organic chemistry was invented. And you have people saying, oh yeah, 90% of the stuff is undruggable. We don't even think about it. So we kind of found that as irresponsible and unacceptable, and been sort of on a crusade, I guess, since then to try and chip away at the undruggable genome.
Very interesting. And I think just for the audience to know, Talus Bio founded in 2020, is that right? And you have some funding as well. So we're going to talk just briefly about who your investors are, how much you raised, and what you're using the money for?
Yeah, absolutely. So we have a nice mix, actually, about 50-50 right now, funded by federal and state grant funding, including this Andy Hill Care Foundation grant from Washington State, and another half of funding from sort of tech bio investors like 50 Years, NFX, and we also were fortunate enough to go through Y Combinator and had investment from them as well. A little bit of a mix, which is a nice way to build a company, especially early on. It allows us to go a lot further with less venture capital at the start.
Yeah, I appreciate that. And you raised $9.75 million seed funding. Congrats on that. And, that's really great to see that resources are being put to good use, because that's pretty staggering. 90% of that transcription, the proteins or targets are never, people think of them as undruggable. So how is Talus Bio going about solving this issue, this problem?
Yeah. So the thing that sort of our fundamental hypothesis at Talus is that there are all these new technologies that enable us to tackle this problem in a different way. And like I mentioned earlier, the sort of traditional approach for drug discovery over the last 30 years has been to identify a target, take that target out of the cell, try and reconstitute that target's environment or its activity in some artificial system, and then find molecules that can block it from working. So instead at Talus, we have the benefit of things like proteomics, which is what underlies our MARMOT platform. And that what that allows us to do is actually, instead of trying to recreate an artificial environment where for these transcription factor proteins that we're going after, these undruggables that we care most about, they only fold and function properly in a native, unmodified human cell. So what we're able to do instead is use proteomics to observe what happens in that native human cell, not have to try and take them out of the cell to study, and find molecules that in that environment where the proteins are folded and functioning properly, find molecules that block their activity and allow us to progress them forward towards the clinic.
Interesting. So, and just for the audience, if you've heard MARMOT and wondering what that is, it stands for multiplexed assays for the rational modulation of transcription factors.
Nailed it.
Mouthful, but very important. I'm sure you took some time to kind of think through what to call it. But how do you actually go about doing this? Is it a computational thing? Is it in vitro, in vivo? What's like sort of that process like?
Yeah, so it's a little bit of all of those things, actually. About 40% of our team is computational. We rely heavily on computation to make all of this possible. I think that's maybe sort of what differentiates these more technological approaches that are unlocking the undruggables in recent years. What it actually looks like on the bench is we have a cell, like I mentioned, a human cancer cell or tissue or whatever the relevant cell type is for the disease that we're working on with a partner or internally. Take that cell, treat it with the molecules that we're interested in. What we actually do is, in an automated way, using sort of robotic systems, we isolate all of the proteins that are actively bound to the DNA inside of that cell at any given time. And what we're looking for are molecules that can go in, bind to these transcription factors, these proteins that are DNA associated and regulate the DNA, find molecules that can bind to those proteins and knock them off of DNA, stop them from doing their job, move them somewhere else in the cell, or ideally to the garbage disposal of the cell. And that's the starting point that we're looking for using these technologies. The computational side of this is all on the back end. Interpreting all of this data and building models about how this works is a big endeavor and what our computational team allows us to do.
Can you talk about some specific examples of work that's in your pipeline now?
Yeah, for sure. So one of the most interesting molecules that we're excited about is a small molecule that binds to this transcription factor called brachyury. This is a protein we've known about for over 100 years. It's one of the oldest sort of developmental proteins that people discovered because animals that lost this protein during their development wouldn't grow a tail. What we realized in the recent years, about 30 or 40 years ago, is that when this protein is turned on in certain cells near the spine, it drives a cancer called chordoma. It shouldn't be on anything, any adult tissue. It's normally turned off very early on in development. But in these cancers, brachyury is turned on, causes the cells to grow and grow and grow. And we've known this for a long time, but haven't had molecules to block this transcription factor, brachyury, from doing its job. So about a year ago now, we identified for the first time a molecule that could bind to this protein. It knocks it off of DNA. When it's off of DNA, it unfolds. The cell recognizes that, brings it to the proteasome, the garbage disposal of the cell, and that protein is cleared and no longer can do its job. So we found those early molecules about a year ago. We've been doing a lot of chemistry since then, optimizing them, making them more potent, selective, drug-like, and even just a few weeks ago now have gotten the first data showing this compound's activity in a patient tumor grown in a mouse and showing really significant disruption of tumor growth in that model.
Well, congrats on the progress you've made so far.
Yeah, thank you.
Yeah. So I guess what stage would you say is this development for your specifically chordoma? Would you
Yeah, so
say,
we're
are you
moving
close
into
to
lead
IND?
optimization now. We've sort of identified a series that has a lot of the properties we're excited about in blocking brachyury. It's pretty selective, and now the goal is to make these molecules more drug-like, improving their pharmacokinetics and other properties. So we'll be ready to put them into first-in-human trials in the coming years.
So I'm sure there are some challenges that you and the team has encountered while doing this. So essentially what you're using is synthetic transcription factors, right? To modulate this protein. Is that correct in a way?
So brachyury is a transcription factor. We're using synthetic small molecules to knock it off of DNA and stop it from doing its job of turning on all the genes that cause the cell to keep growing.
Are there any unique challenges in that approach?
Yeah, I mean, one of the big things that we need to be aware of, especially for transcription factors, is this idea of selectivity. So brachyury, in some ways, it's an ideal target because it's not active in any of the normal tissues in our body. So as long as we're hitting that target really specifically, there shouldn't be any toxicity that we don't expect. But that's not true for lots of other transcription factors that are implicated in cancer and fibrosis and inflammatory disease. They can have activities in other parts of the body. They're regulating genes in those tissues, and understanding those networks is really important as we think about bringing a molecule forward for some of these other targets.
Is that one of the reasons your team has selected to go forward with brachyury as opposed to maybe some of the other drugs in the pipeline?
Yeah, so as a first program for us as a small biotech, we prioritize a few things that brachyury kind of checks all the boxes for. It's a huge unmet need. So these patients that have chordoma, this rare sarcoma driven by brachyury, don't really have any other treatment options from a medical point of view. They get surgery and radiation. As you might imagine, a spinal cord tumor is pretty difficult to operate on. Essentially what happens is they wait for recurrence and do it again. Chemotherapy isn't effective. There's no targeted therapies or immunotherapies that have shown to be effective. So really bringing forward this molecule efficiently is really important from that point of view. And luckily for us, the Chordoma Foundation's really been leading the way and laying the groundwork for what it would look like to do a trial with these patients, with this relatively rare patient population. So the unmet need, the biological rationale, like you were mentioning, as well as the efficiency in which we can get clinical data for these molecules is sort of all the things we're thinking about right now in prioritizing this program.
I appreciate that. And, I'm switching gears a little bit here, but in the biotech space right now, everyone's sort of talking about the use of machine learning and AI to assist with drug discovery all the way down to patient recruitment for clinical trials. So, and I understand that, there's a component of computational and AI in Talus Bio. Can you kind of describe specifically how you're leveraging AI?
Yeah, one of the things, at least that we're seeing and starting to realize from this most recent revolution in AI is that it's not the model.
I like how you said most recent evolution, because there has been many sort of iterations of, AI is huge. Even in the 70s, like there was a lot of hype about it. So this is just the current hype cycle for AI and arguably the biggest one, right? So let's see what happens. But sorry to cut you off there.
No, it's true. It's true. And that's the sort of danger, right? The hype, overpromise, and then when things don't work out as planned, it discourages innovation and investment in the area. But what we've been seeing is that all these new models are great, but what makes them great? Yes, the architecture is really important, but really they're nothing without the data. LLMs are only working and coming to fruition now because they have all this data, whatever, Twitter, Wikipedia, to pull from. From a biotech point of view, we, with the approach we're taking and using proteomics, protein sequencing, every time we test a molecule, we see how that molecule affects every single protein in the nucleus of that cell. So very, very quickly, we're amassing this massive database of protein dynamics, gene regulation, and it's really the only database like this that exists in the world, to our knowledge at least. So that data is really the core component of our AI engine. We can ingest that all in, understand what chemistry leads to what responses in this regulome, this genome regulatory proteome. And then after we build those models, we can actually start predicting, well, if we want to hit this transcription factor or this undruggable target, what types of chemistry should we be looking at to try and modulate that in a specific way? And then we make those molecules, test that hypothesis, and start spinning this flywheel around faster and faster and learning as we go, both with the models in hand.
Have you been able to sort of identify databases, public, maybe even public databases or private, that you've been able to include in your data lake for your work?
Yeah. So the lab I worked in here, you mentioned the Altius Institute in Seattle. That's where I did my postdoc. We were one of the labs that was deeply involved in the ENCODE project at the time and other projects like Roadmap. These were epigenome profiling consortia where the goal was to take every tissue in the human body, profile it with a suite of assays to essentially figure out where all the genes are, when are they on, what proteins are controlling what genes are on. And all of that data is very valuable, but it was very targeted. And you kind of had to know what protein you were looking for at the beginning before doing those experiments. So we leverage that data. It's all really important, but we're kind of taking it to the next step now, allowing us to, the way we do things now is completely unbiased. So we don't need to pick a certain transcription factor at the beginning. We can actually look at everything and then allow the machine learning or the AI models to figure out what's actually the important thing going on here rather than trying to guess at the beginning.
I appreciate that answer. So given that you have a couple drugs that you're working towards, or treatments at least, in your pipeline, and you mentioned how the treatment for chordoma that you're working on is in lead optimization, are there any specific types of partnerships that you're looking for from the community now for Talus?
Yeah, so I mentioned the models we're building. The sort of scale we've gotten this platform to now has led to, in some ways, sort of an embarrassment of riches for us at the moment. We have profiled all of these molecules against all of these transcription factors, and we now have dozens of these molecules that are modulating undruggable targets that we know are important, that we don't have the bandwidth or the financing to prosecute ourselves. So what we're really looking for are folks with deep expertise in the biology or the chemistry with targets that they know are important, but have been sort of put on the shelf because of this undruggable idea. So we have all these molecules we're willing, able, and ready to partner, and also offering access to the technology itself to advance these molecules as quickly as possible with folks that have built out the expertise in the sort of deep disease biology.
Yeah, and I hope someone listening out there or watching this now who might be thinking, oh, wait, I can maybe help with this. Feel free to reach out to Alex or myself on LinkedIn. We'll be able to, get you to the right person.
Yeah, absolutely.
Because I think that's important now, like getting the right folks involved in specific projects, connecting them to the right team. That's the hard part. Once you're connected, you sort of have this ability to get energy or create this sort of environment where things start to move really quickly, especially with the technology we have now. It's quite amazing.
Yeah, absolutely. And I know I've had this experience, and I'm sure lots of people working in biotech and pharma and academia have too, where they look at their lists of their best targets and it's like, oh, this is a transcription factor. What's next on the list? We can't really go after that. Reach out. We might already have a molecule for you. So let's see what we can do.
I appreciate that. So right now, I guess one thing I wanted to ask you, first of all, are you currently raising another round for the company?
Yeah, we're wrapping up a raise. We're raised now to really help move that chordoma program forward really efficiently, expand our pipeline out a bit more, and continue to keep building these models and these data larger and larger. Yeah.
Awesome. What's your take on the more general biotech economy these days? Like, what's funding looking like from your perspective as you've been talking to investors, LPs, things like that?
Yeah, I mean, I can give my perspective. It's probably not the best takes for one company going out there, but it's hard. You know, it took us 6 months or so to get to a point where, you know, we're getting close to closing the round that we need to keep our science going. But you know, we're in it for the long haul. The ups and downs in the market are going to happen. It's our job to, whether it's through grant financing or partnerships or other creative funding sources, it's our job to keep the science going forward and make sure that we can deliver these molecules to patients as quickly as we can.
And we hope you continue to do that. I think it's important for entrepreneurs like yourself to really push hard on this because, like you said, it's not easy. I hope that 2024 brings in greater deal flow for you and other companies as well. And it seems to be the case, at least from the people I've been speaking with in the last month or so. So I hope
everyone should keep saying it. Let's all agree to just.
If we all believe it, it must be true, right?
Exactly.
So Alex, I just want to thank you again for taking the time out of your day to speak with us. Is there anything else you'd like to share with the community, potential investors or partners out there?
Just, you know, thanks, Ray, for having us. We really appreciate the chance to get to share our science and connect with the community. You know, we're really focused on one really small sliver of this undruggable problem, but just, you know, if other folks are interested in tackling some of these other parts of the undruggables, connect with us. It's a huge problem. It's probably the most important problem in therapeutics for our generation to do. And happy to help and help this mission any way we can.
And Alex, we're happy to help as well. There will be links in the show notes to your LinkedIn, your website, and everything like that. So if anyone's just listening in on Spotify or something, you could check out the show notes for more information. And yeah, if anyone has any comments or questions, please leave those down below and we will get back to you. All right. Thank you.
Yeah. Thanks for having us. Cheers.